Hey, there we go. Okay, now we started recording. Missed the Slim Shady introduction. Okay, folks. So I'm just going to Ross. I am just going to make you. If you could just admit people for me, Ross, would that be okay? I'm just going to make you a host as well. If you don't know him, Ross, our Saskatoon lead, we'll talk a little bit more about him. He's going to be a co-host as well, and Scott Wells is here, also hosting. We'll get started here. So again, everybody, welcome back. We really appreciate you all taking, we took a little hiatus over the summer, but we are happy to say that we are back. We are ready to go. Can you make me co-host, Ross? Sorry, I somehow gave up host when I made you host, so apologies. Hopefully we're still recording. And y'all can see my screen? No issues? Awesome. Okay, so again, welcome back. For those folks who are new, this is Data for Good Saskatchewan, newly Data for Good Saskatchewan. Previously, we were Data for Good Regina, Data for Good Saskatoon. We did merge, and right after the merge, we took a little hiatus. So please make sure that if you're still on the Saskatoon group, we won't be posting there anymore. We still have it up there for legacy purposes and to help folks migrate, but just make sure that you sign up with Data for Good Saskatchewan at the link saskatchewan.dataforgood.ca. If you don't know us, we're a group of data and AI enthusiasts from Saskatchewan looking to really build community, both in the data space, but also build community more globally by helping the nonprofits in our province with any kinds of data challenges they have. We've got experts, they got people who need help with data or AI, and we're here to help solve their problems. So if you don't know what we do, we run lots of projects to help the nonprofit sector with any kinds of challenges they may be facing. Usually it's more on the data side, just because that's where they're at in their data journey. But as we've been expanding and as we've been growing, we are looking for a wider variety of projects as well. So just some past projects we've done. We've talked about these ones before if you've been to one of our talks. If not, you'll see that there are a number of nonprofits we've helped out, such as the Sexual Assault Services of Saskatchewan, Regina Public Library, Sask Rugby, Regina Food Bank, and we are also behind the nonprofit data accelerator program that you might have seen us posting on Facebook, on LinkedIn and on Meetup about quite a bit. So really proud of what we've done over the last year. We've done made a lot of efforts to really expand out our membership base. And moving into the new year, our new season here from September until next summer, we have a really exciting program of events and program of activities that we really want to share with you folks. And we're hoping to bring you all along in the journey as we expand and as we grow. So just to call out our leadership team here, previously it was myself and Scott Wells doing a lot of the work here, but we've really expanded out our leadership team this year, adding a couple of key folks here. And sorry, I also mentioned we also had Chuck Plant from Saskatoon and Josh West over in Calgary helping us run the website. But we have added our Saskatoon rep here, Ross Gadzella. You might have seen him at a talk if you've come from one of Ross's talks up in Saskatoon, and you saw him hosting the Saskatoon part of the Data Accelerator program when we ran that event. We also have Jasmeet Singh, who's helping us on the marketing side, and then Biaka Amia, who's also another one of our dedicated leadership team up in Saskatoon, working to really expand and grow our project base up there. One thing you are going to see from us for this next little bit is that our big focus and big push is going to be getting more activity and more events and more projects up in Saskatoon. So you will see a lot more of that. And I think I dare to say Ross may even be taking my place at the next one of these. Hopefully he has a better taste in music than I do to share with you folks. So I want to just highlight, before we get into our actual talk, I just want to highlight some things we got upcoming. We have been working really hard to get a lot of activities that we can get engaged with and that we can get our members involved with. So next month, we will have a talk from Better Cart Analytics. What's the name of our speaker, Ross? I didn't catch the full one.
It's Melanie Morrison. She's the CEO.
Yeah, Melanie Morrison, the CEO of Better Cart Analytics, will be coming to talk. We're still ironing out the date there, and we have some other talks in the pipeline that we're working on. We are also planning a new sort of training program for some of our junior members called the Data Cultivator Program. It's kind of like we noticed with the Data Accelerator Program that there was a lot of need to get folks up to a certain level before they're ready to really take on and lead a project. And so we are laying the groundwork to roll out a data cultivator in the near future here. We don't have a firm date just yet, but we are putting things together behind the scenes there. And the hope would be that it will enable more people to run the data accelerator program, and even make it a little easier for us to run so that we can focus on some of the leadership pieces down the line, because we know we'll have more experienced folks who can take on leadership roles in that program. Ross and the rest of our Saskatoon leadership team, shout out. They've been doing a great work up there. They've got a new project that's cooking, got it in the pot with the Saskatoon Food Bank and Learning Center. So we're just finishing up and revising some of the, or just working on the actual statement of work for that one, or the actual agreement. And so we're working on that one. We'll have more information about that soon. Once that's ready, we will put a call up to the membership to see who's interested in participating in that project. Our priority will be getting some Saskatoon members involved in that one, just because we are trying to focus on broadening out our Saskatoon base. But again, if we'll be open after that, we'll be open to any kind of membership. We do have dates for the Data Accelerator program. We don't have them fully locked in, but we do know we're going to be running our next Data Accelerator program between February 2025 and May 2025. We've been really hoping to run another one in September, but it's a lot of work. It takes a lot of effort to put these things together. So we figured let's just leave it to one for this year. And then if it expands, we can look to grow it even further into other regions and other delivery methods into the future. And then last but not least, we are going to have a booth at the Reimagining Nonprofits Convention at the University of Regina on October 19th and 20th. So what we'll be doing is, for some of our members who've participated in projects and who've participated in the Accelerator program, we will be putting a call out for folks to look for folks who might want to staff that booth. Because we will have a booth there where we can advertise what Data for Good does. And, myself and Scott will at least be there. We're hoping that we'll be able to get a few other folks who want to join us and have some fun. So, Scott, you want to say any more about that? Because you've been involved with that a little bit.
Yeah, I think the really neat and interesting part about this is the nonprofit community has actually reached out to us to say, can you come to this? We'd like to have you there. A lot of the organizations are really interested in moving into kind of progressing and maturing their data journey, I guess. And so they really want to have us there. And so it's going to be fun and interesting. And for folks that kind of come out with us, you'll be able to really get a sense of what's going on in the community, what the needs are, and where these nonprofits, charities want to go in the future. So it's going to be a lot of fun.
Right on. All right. I won't keep you waiting any longer. Joining us today, we have an awesome talk from Tayab Soomro. And fun fact, Tayab was actually one of the original participants in the Accelerator program. I think he was like driving his car back from Saskatoon back and forth. And we said, man, you're so hardcore. And so we said, you know what, we'll get something going in Saskatoon in the near future. So we're still, I know you're still waiting on that, but we appreciated your support in the beginning there, Tayab. Just to introduce Tayab, Tayab's career has taken a unique and fascinating path, leading him to a title not often heard in everyday conversations, bioinformatician. In this role, he dives into the complexities of biological data, uncovering insights from the murky depths of genomics. Tayab holds an MSc in agricultural genomics from Dalhousie University and a bachelor's with honors in bioinformatics from the University of Saskatchewan. His expertise has led him to provide bioinformatics consulting to the government, helping him to unravel genomes of various crops that influence the traits that we see in the field. Beyond this, Tayab is the co-founder and CEO of PathoScan, a startup working to bring fast and accurate crop disease testing to farmers directly. His work is all about merging technology with agriculture, using diverse data sources, and providing meaningful insights into a sector that's crucial for feeding the world. So today, Tayab will be sharing his insights on how PathoScan leverages data to tackle agricultural challenges. Tayab will also explore the various types of data integral to biology and highlight how bioinformaticians play a crucial role in data analysis in the field. With that, Tayab, take it away, my friend.
Awesome. All right, so I guess just share my screen here. Okay, so are you guys able to see the screen? Perfect. Thank you so much for the introduction, Ryan, here. And yeah, I'm really excited and stoked to learn that Data for Good has some representation in Saskatoon. Really excited to see what comes out of that. Yeah, so Hi, everyone. Thank you for agreeing to listen to me. I'm, sort of excited to talk about bioinformatics, which is sort of an odd topic to talk about at a data science conference. But if you bear with me and give me some time, I will try to make you appreciate the challenges that bioinformaticians face, the data challenges, data visualization challenges that we face in the world of data science. So I'm going to talk about bioinformatics, data science, and ag-tech, and sort of the circuitous path that has led me to this fascinating field. And as we sort of go along, I want to make an effort to sort of make you appreciate the importance of bioinformatics and the unique challenges that we deal with in terms of the data. So first of all, I think it's sort of an easy place to start is to understand how did I get roped into all of that. So I started my undergrad in computer science, hoping to one day become a software engineer. But during the summer of my sophomore year, I got an opportunity to work with Dr. Matthew Links, who was a research professor in the Faculty of Agriculture in the Department of Animal and Poultry Science at the University of Saskatchewan. Now, you might be thinking, what does a computer science student have to do with agriculture and biology? Well, that's exactly what I thought when I accepted the position. But as a student, you get any work placement, it's a blessing. So I just went for it without thinking much of it. So what Matt wanted me to do was develop visualizations, basically creating these plots that overlaid some signal data with DNA sequences. Honestly, at the time, I had no idea what this all meant and what I was doing. And so he would often talk like a million miles a minute about biology, DNA, sequencing, all that stuff that I knew absolutely nothing about. I kind of felt out of place, but I decided to sort of take this as an opportunity to write some code, learn data visualization, and those types of things. But if you want to learn a little bit about that, you can actually check out that story that was published at the University of Saskatchewan website. But as I sort of worked on this project, I started to learn a little bit more about biology, started reading research articles, various topics that were way beyond the scope of my studies at the time. But I realized that biology was, way more than dissecting frogs and mixing chemicals in a beaker, which honestly was my impression at the time. There's this entire new world of data that needed to be explored, understood, and analyzed. So I started reading papers about computational biology, and I even attended like conferences where researchers were using data to make impactful discoveries in agriculture. And so I realized that bioinformatics actually involves statistics, data science, biology, biostatistics, and things like that. This was very interesting to me. So after that summer project, I decided to switch my degree from computer science to bioinformatics, which sort of felt like a perfect blend because it involved computers, which I was interested in, but also biology, which was my newfound interests, if you will. And 8 years later, honestly, I think switching to bioinformatics was one of the best decisions I've made in my life. Although it's sort of a bit tough when you're trying to introduce yourself, like Ryan was doing just a moment ago. Usually when I'm at a party, I'll just say I teach plants how to use computers. So. Over the next few summers, what I did was I worked in the same lab, but on different projects. And I was learning so much about bioinformatics technologies. So finally, I learned what DNA sequencing actually is. In our bodies, DNAs are sort of coiled up in a thread that unspools when the body needs to make proteins. And when the body is done making proteins, that DNA then spools back up and it's stored away. But sometimes proteins are not made correctly. There are some errors in protein translation. So, for instance, prion disease, which is a fatal brain disease that is caused by misfolding of these proteins. So, when proteins misfold, it has some effect, downstream effects in the brain, and that causes people to die. And it's also known as mad cow disease, which has a huge story about the beef patties and stuff like that. So understanding how the DNA creates proteins is really important, right? But the first step to do that is to read the DNA. What actually does it contain? And DNA sequencing is a method that allows us to do that. And how it works is we first unspool that long thread, which is about like 3 billion base pairs for humans. So 3 billion letters long string. and you cut it down into many pieces to make it manageable, because no machine can take up 3 billion, a thread of 3 billion long and read it on the other end. So you cut up into these pieces, and then these pieces are then passed into what's called a DNA sequencer, which takes each letter, each base, and it records it onto the computer, turning that DNA into sequences of A's, T's, and G's, and C's. So adenosine, thymine, guanine, cytosine. But these sequences, you have to understand these are like in millions of pieces, right? So these are all these different fragments that we have. Now, there's this challenge of putting these pieces together, so stitching it together to understand what proteins they code for. And so now, you can imagine the huge computational challenges that exist in piecing these strings together. And this type of computational challenges have actually led to some of the most sophisticated algorithms that we have. So one of the algorithms is called De Bruijn graph, which essentially is if you know a little bit about graph theory, there's these nodes and edges, and it usually, what it does is it puts these fragments into nodes, and it tries to connect them with edges and to essentially assemble those pieces together. And so one of the ways that DNA sequences reads these bases, so A, T's, G's, and C's, is through what's called like electrical signals. So if you imagine there's a membrane with a lot of pores in it. These pores are just big enough so that a single DNA thread can pass through it. And this membrane is also electrically charged. And we know that DNA is negatively charged. So when we apply electric current through this membrane, it kind of creates like a pull for the DNA to go through these pores. And as it passes through, each of the unique bases, like A's, T's, and G's, creates sort of like an electric current, and it's different for each base. So A creates a different electrical current than T. And based on that, we can actually see what the base is, and we can resolve the bases. But obviously, with biology and everything is very complicated. The signals that we get are very noisy. They, in their raw form, we cannot really detect what the bases are. So we have to use statistical methods, machine learning, to sort of interpret these bases. And so coming back to my first summer project, when I learned all of this, it all started to make sense. So what I was doing essentially was I was plotting these different signals that were coming out of that DNA sequencing machine against the bases that were resolved, that you can see right here. So the objective was to look at the signal data and to see how accurately is the machine learning algorithm detect or resolving these bases. And this process is called base calling, which means that you're sort of calling what these signals resolve, what base it resolves to. We know from this technology that the error rate for resolving these bases is really high. So the overarching goal for that research that I was working on was to do some sort of transformation on these signal data to clean up the noise, and then so that we can, when we resolve the bases, it's a little bit more accurate. And so that's sort of what the project was. And so this gives you a general wave of how data visualization is used in biology. But here comes the first data visualization challenge in bioinformatics. The signal plots that you see, so imagine so remember back to when I mentioned that we have the human genome has 3 billion base pairs. Well, each of these points here are about 100 million data points that we have to sort of visualize. And not only that, we have to make them interactive because we want to explore a single event, signal event, and see how it resolves to each base. So this is massive amounts of data that needs to be visualized. So the usual tools that we use in data science, like Jupyter Notebooks, Matplotlib, or Plotly, they just can't handle this type of data. So they either crash or they take forever to load, which is not useful. So what this type of data analysis has allowed for is sophisticated, memory efficient ways to actually visualize these types of data. So there are various tools that we use that are different from the general data visualization techniques that we are all too familiar with. So that's one of the challenges that bioinformaticians face. And I hope that this sort of give you an appreciation of the data visualization challenge, but there's a whole bunch of other data analysis that bioinformaticians do. And one of the other data analysis that we commonly do is called population genetics, where we explore the genetic diversity that exists in a population. And so that's what I worked on next. So when I finished my undergrad, I got an opportunity to pursue a master's degree at the Dalhousie University in Nova Scotia. So I was there working under supervision of Dr. Sean Miles, who had gathered over 1,000 different apple trees into a single orchard. And the purpose for that was to understand the genetic diversity that exists in all of the apples that we have in the world. But when I visited this orchard, I was just fascinated by the different kinds of apples that exists. So there are apples that have black skin. It's called black Oxford. And there are ones that have pink flesh. They're called pink pearl. And so there are also apples that like taste like candy and and things like that. It's just so unreal the amount of genetic variation that we have in apples. And when you sort of smell these apples, all of these apples have very distinct smells.
I mean, took the member, right? I mean, took the.
Sorry?
Sorry, just I think some crosstalk. Apologies.
Oh, okay, no problem. And so yeah, so when you look at the smell of these apples, they all smell different. So I wanted to understand the genetic underpinnings of the aroma that we experience in apples. So like what is inside that genome that orchestrates these different smells? And so first of all, how do we go about doing that? Well, we know from the biochemistry that the smell or the aroma is essentially these the volatile compounds that bind in the back of our nose to these receptors that we have. And depending on the number or the types of compounds, these volatile compounds that bind, we experience different smells. And we have over like 300 different compounds that we have available. And so there's these fancy machines that can extract those aroma compounds from apples. And this process is called gas chromatography mass spectrometry. So essentially, the idea is that you take a piece of apple and you crush it up and make it into a solution. You electrically charge that solution and you run it through a column. It's like a long column, and based on the mass of each of the compounds that come out, at the other end, they will reach the other end at different time points. And based off of that, you can determine what compound it is. It's very complicated, but it works. So at the end, what you get is you get this list of compounds that are present across these different apples. So one of the first things that you can do is figure out what their abundance and ubiquity is, like how many different compounds are there, first of all, and how much of a specific compound is present in apples. Well, that's exactly what I did here. So each plot that you see in this plot represents an aroma compound. So butyl acetate, hexyl acetate, ethyl butyrate, 2-hexanol, things like that. The x-axis here represents the total, the ubiquity of the compound. So how many apples this compound is present in. And here I've taken the 500 apples. So there are compounds that are present in all of these 500 apples. So which is very interesting. But a compound can be present in an apple, but it may not be present in high amounts. It may only be like little amount of that compound present. To capture that, the y-axis here represents the abundance of that compound in the apple. So for each given compound, I have how many apples it's present in and how much it is present in that particular apple. So you get this plot here. And each dot again is sort of the aroma compound that we're looking at. So one of the main observations here is that the dots that are at the bottom left here are they're sort of the unique compounds that are only present in a few apple cultivars, and they're not very abundant, right? They're very small quantity, but they're also not very abundant. But when you contrast that to the dots that are on top right, then these are the compounds that are present in across all the apples and in high amounts. So, that's sort of the first observation that you see. But what are the major takeaways here? The first thing that you notice here is that butyl acetate is sort of it's twice as much abundant than the next compound, which is hexyl acetate. In fact, it's sort of when you look at the compounds that I've labeled here, these make up 98% of the total aroma in all the apples that we have in the orchard, which is actually really really interesting, because these are the small number of compounds that make up the apple aroma. So when you smell an apple, this is what you're, sort of, this is what's binding to your noses. So now here's an interesting thought, coming back and bringing it to the data science aspect. You go to an orchard, you take an apple that is very distinct in its smell. You've smelled nothing like that before. The chances are, when you look at that apple, it's going to be somewhere around here in that plot, right? Because it's very distinct. It has different smell. So what we've done here actually is we've taken a whole bunch of seemingly nonsensical data, and we've created a hypothesis testing tool that the subject matter experts like biologists and breeders and things like that can use to understand the biology of apples. So, the data analysts in the room, they're all too familiar with this type of thinking and analysis, right? We create hypothesis testing tools with statistics and visualizations. This is exactly what bioinformatics is, right? It's the manipulation of data, or sorry, not manipulation. It's sort of the representation of the data that tells a story, essentially. So another common analysis that we do in bioinformatics is called principal component analysis, which, in this case helps us explore the apples, how the apples are sort of related to each other in terms of their aroma profile. So if you think about the structure of the data that I'm working with here, we have for every apple about 3,000 different compounds. So coming back to the machine learning terms and data science terms, we have the feature size of every sample about 3,000. So this is a highly multidimensional data. What PCA does is it reduces the dimensionality into a few components that explain most of the variance, which exactly is the point, right? We want to look at the apples that are really, really different. So there are compounds, there are chemical compounds, aroma compounds in apple that contribute very little to the variance of the overall apple, right? If there's only like 0.0001% of that compound present in the apple, chances are it's not really important. So what we do is we then sort these compounds based on highest to lowest variance. And then those are sort of shown in principal components. So the first principal component, or PC1, explains most variance. Second principal component explains second most variance, and so on. But generally what's done with PCA is we plot PC1 and PC2. So we plot the most variant axes and the second most variant axes, and that tells us something about the relatedness of the samples in terms of their aroma profile. So the two samples that are close together in this plot, and each of these dots, by the way, are apple cultivars. The two samples that are close together have similar aroma profile. So if you take two very close apples, and when you go out into the orchard, you smell them, chances are they're going to smell pretty much similar. And then the apples that are sort of further apart with each other, you look at the pairwise differences, they're completely different. Their aroma profiles are very different. So you'll notice here also that I've colored these apples based on their harvest date, which essentially is the time when the apple is ready to be harvested, and it was harvested from the tree. We see that there's a correlation between the harvest date, which essentially translates to the when the apple ripens, to actually to the aroma profile, right? So for example, apples that are harvested early, which are sort of the yellowish, are far left on PC1, and apples that are harvested late, which are sort of the darker colors, are to the right. So PC1 seems to explain the harvest date of the apples, and so when you look at what that means is you look at the volatile profile of an apple, or the aroma profile in an apple, that actually is explained by its harvest date. So, jumping ahead a bit, early harvested apples tend to have less volatiles versus late harvested apples, and we're going to get to that in a second. And obviously these you also see some outliers of the apples. When you look at these apples that are different from all the apples here, you'll definitely see that these apples are different in their smell. And when we actually went into the field and looked at these apples, they were crab apples. They were like apples that were not they didn't get into the breeding programs and things like that. So they actually are very very distinct. So what we learned is that time it takes for apple to ripen, which is captured by its harvest date, is associated with apple's aroma profile. But how does the aroma differ exactly between early and late harvested apples? So to answer that question, what we did here is we did correlation of apple's harvest date with the number of compounds that it expresses, right? So take an apple, the number of different volatile aroma compounds that it has, you try to, do a correlation. It's essentially a linear regression analysis that we've done here. And so on the x-axis, we have the harvest date, and you can see it's a nice spread between sort of August and November. This is Julian days, by the way, which is, the counting 1, 2, 3, the days from the start of the year. And then on y-axis, we have the number of volatiles that are detected for a particular apple. So each dot here is an apple. And we can see that there is a relationship. Early harvested apples tend to have higher number of volatiles, and late harvested apples tend to have lower number of volatiles. But when you look at the abundance, so how much of that volatile compound is present, it's also true. Like the early harvested apples tend to have not only high number of compounds, but high amounts. And so that's very interesting. But what that means practically is, if, let's say, you imagine going to an orchard and you're smelling like a ripe Gravenstein, like, one of those green apples. These are early harvest apple varieties. You'll notice that these apples have a lot more aroma than something like late harvested apples like Honeycrisp. But, and so now that's sort of that's what the analysis of data gets us, right? So it gets us to this story of early and late harvested apples. Now you can actually take a step further from there and understand the biology of it. And that's sort of where the subject matter expertise comes in. So what I did then is like, okay, thinking about this relationship, and then looking at the understanding the metabolomics, the biology of apples, and understanding what it exactly it means. Well, it turns out the reason why we see this relationship is that when apples are harvested, they are put into 1 month of cold storage. But when they're put into cold storage, early harvested apples have different sort of cycle metabolic process, right? Because they are harvested early and they're ripe early. So when a month for early harvested apples is different than a month for late harvested apples, and that's why we sort of see these differences in the smells. And so, if we're getting anything out of this slide, it is that, it's the basic linear regression that we're all too familiar with that tells us this story. And linear regression actually is used quite a lot in bioinformatics. So I want to pause here a bit and just, give you some time to sort of appreciate, how we turned a bunch of biological and biochemical data into a nice story that tells us about the metabolism of apples. This is sort of very similar to what data scientists, and how they tell story from their complex data. So, Ryan mentioned one of the projects that Data for Good did was understanding the donation patterns of Regina Food Bank, right? So there are some data that was collected by the Regina Food Bank about, the time that the donation happens, the demographic of the donors and things like that. We can actually use that data. We've, Data for Good team use that data to gain insights on donations to help with creating, outreach and programs that increase the donation amounts and the frequencies. That's similar type of statistical analysis that we do here.
The story we all want to hear, Tayab, is what's your favorite species of apple or favorite type?
Something that you don't get here. So at that orchard, we had this apple called I forget the name of it. Oh, forget the name of it. It's not very tannic, but it's got a little bit of a spice to it. And I'm sure it's going to come up in my head, and I'm going to tell you the name. It's the best apple that you can get in sort of the eastern Canada. I haven't found it here, and nor have I found it in BC. And Briosa and Honeycrisp are pretty common, but yeah, if I find that's probably the best apple. Yeah,
we're missing out.
We are definitely. So, initially I talked about, population genetics, but up until now I've been talking about just population. So let me tell you a little bit about the genetics of apples that I worked on. And, there's a lot of data science and statistics that goes into the genetics as well. So, every apple, as is with us or any living thing, we have DNA, right? And it's sort of the blueprint of, how things change and what makes us unique, right? There are changes in the DNA, like A's, T's, and G's and C's that makes me me and makes Ryan Ryan, right? Like there's these differences that make us unique. We call these differences polymorphisms. And you look at the reference, some reference, and you look at all the other different humans or apples, they're all different from the reference and amongst each other. And that's sort of what makes them unique, right? So Honeycrisp and Gala, the differences between them is the changes in their DNA. So at any given location in that DNA, the two apples can have different bases, right? So for example, most of these apples have A, some of them have T, and that's sort of the differences that are called polymorphisms. And that those differences contribute to the unique phenotypes that we see. So what I did here is I actually mapped out all these differences across different apples that I studied. This is sort of the, we took the 250,000 polymorphisms, which is like 250,000 of these differences across 1,000 different apples. And again, so you can imagine the dimensionality of this data that we're working with, right? 250,000 data points across 1,000 different samples. So this is a huge data set. And again, it's a huge data science problem. So, obviously there are sophisticated data structures that we have to use and data file formats to store such a data. And so this is just a quick example of how this type of data is stored. We have each of the position in the genome, and then the sample 1, sample 2, sample 3, we quantify it based on 0, 1, or 2. I'm not going to get into the details, but essentially you appreciate that, for this is sort of the transfers. The features are on the as rows, and the samples are in the columns, and you use this data set to sort of this data structure format to work with genomic data. So one of the questions that you can ask with the SNP data or this polymorphism data is what regions of the genome are correlated with certain phenotype or the aroma compounds in our case. And so, what you do is you look at each of the single mutations in that genome, and then you try to create a linear model of that mutation and across the phenotype that you're interested in, which is a list of compounds. And so you look at the association of that trait. So, this type of analysis is called genome-wide association studies, in which what we take is we take every polymorphism and look at its strength of correlation with the trait. So just to give you a visual representation of how that works is across your 1,000 apples, you measured the concentration of, let's say, a compound called 1-hexanol, across your 1,000 apples. And then you have, so you have two vectors. You have the SNP vector across the apple, and you have the vector of 1-hexanol concentrations. You try to create linear correlation between these two vectors, and you get what's called a p-value, right? That's sort of a standard that you get from linear model, and it sort of indicates about the strength of that correlation that you see for each polymorphism. So you do that, about 250,000 times for each SNP, and then you plot that p-value in this plot here. So this is sort of the linearized genome. So you have every chromosome here. Apple has 17 chromosomes, and you sort of divide each SNP on the chromosome, and you plot the value here. The y-axis is the p-value, but it's actually log 10 transformed. The reason why we log transform is because it makes it for easier visualization, and you see the associations that are really striking right off the bat, which we sort of see here. So the strength of the correlation is denoted by the y-axis. So the higher up the point is, the SNP is on the y-axis, the more strongly associated it is with a trait. And then the line here, it's essentially just a thresholding that we do to remove any false positives and any associations that are not really strong. And so we see that one striking association, sort of comes up, and that's sort of what you want to see in a GWAS plot, which says that on that chromosome 3, there's a location in that genome that is strongly associated with the changes that we see in hexanol, right? So that tells us about first of all, then you explore what is inside that. You look at the genome, what is there that may cause. Then you look at the genes that are around there, and so in this case, we found this gene called NAC 18.1, which is a transcription factor that actually is related to producing these volatile compounds. So a nice story about finding a causal variant that causes changes in a biological compound. And so yeah, so this is an example of the type of data analysis that you do. And so this is sort of my master's. I finished that, and then I came back to Saskatoon. And here I met Ethan, who was the co-founder of the company that we formed. And he had thought about this technology that could be deployed into the farms for crop diagnostics. Right. So, I'd worked with plant pathogens in my undergrad. So therefore, when he sort of talked about this technology, I was really excited. And we jumped on board and we created a company that we called PathoScan. And so it's been a year and a half since, and we've been doing pilot projects across Saskatchewan, and it's really exciting. So in the few minutes that I've left, I think we're just about time here, but I want to talk a little bit about how it works and what is exactly is it that we're doing. So what PathoScan devices allow farmers to do is test for crop diseases right in their fields. So traditionally, what farmers have to do is they send the samples to a lab facility that's like 3 hours away, wait for the results to come back, and by the time they get the results from their from the lab, it's already too late for them to do anything because the crop is dead. So what we developed is this device that they can run, in their cars, in their offices, in the field, that can do a biochemical reaction called PCR. And we're very familiar with PCR from the COVID days where we had to take that PCR test before we could fly. Right, so similar stuff, but we just shrunk it down into the size of a box, and we're able to do it remotely. So what a farmer does is they place a piece of a plant into this tube. There are a bunch of steps to get that tube ready, but once it's ready, you place it into one of these wells. You close the lid, press a button, you start the reaction. In about 20 to 30 minutes, the reaction works, and at the end you get a nice report that tells you about whether or not the disease was present and whether or not you need to spray. So I want to briefly touch about the type of data analysis that we do to get this result, and it is basically over time as the reaction is happening, there's color intensity change in the tubes, and we captured that using statistics and machine learning, and based off of that change in some statistical significance analysis on it, we're able to confidently say that the color change is indicative of presence of a pathogen, or the color change wasn't significant, so that pathogen is not present. So there's quite a bit that still can't be shared here just because there's work being done. But this is sort of the general idea that we use of the data that we play with in PathoScan. And I think we're at time, so I'm going to stop here. But I hope this was interesting and not too boring. But I'm happy to sort of answer any question, bearing we have time.
Nah, this is super cool. Although I've taught statistics, so I mean, I guess I'm a little biased. But no, this was awesome. Thank you so much, Tayab. We really appreciate it. We've got a few messages that have come through, just some questions that you have. And yeah, well, hopefully you can enlighten us. So Lydia is just asking here, just in the chat. She's just assuming this explains why fruits in the farmer's market. This was to your apple context here, and just assuming this is why fruits in the farmer's market have a stronger aroma because they're harvested late. Is there insight to that? Is there any idea on that, or does the data say anything about that?
So over time, so what happens is when you eventually see the fruit in your market in the supermarket, what's happened is it's first of all, it's gone through the cold storage, but it also has traveled quite a bit. So and it's gotten heat and stuff like that produces. So one of the main ways why we experience smell in apples is through ethylene production that happens in apples, and that's sort of what makes any fruit ripe, right? So when you look at mango, higher ethylene production means that they're going to ripen quickly and things like that. So when you're seeing these apples in supermarkets, they've gone through the stores, they've produced a lot of ethylene, and at the end when they come, they're like they have a lot of aroma. But the aroma that we were interested in is when the apple is like right off the tree. Because when you go for apple picking, you actually notice these differences. For example, next time you take Gravenstein from a plant and then take a Honeycrisp, you'll definitely notice the aroma difference.
Right on. Well, thank you. Appreciate that. And I was gonna just picking up on my own interest. I come from a farming background, and my brother still farms, and I'm kind of curious. Like, is the farmer the target market for this one? Like, is that who you're aiming for with the boxes, or who are you looking to go after for your product?
Yeah, so farmers eventually are the folks that make the benefit from this, but most of the crop disease testing is happening by agronomists, which are sort of the contract consultants that farmers hire to help with farming questions. So these are sort of the folks that we are targeting currently.
Cool. I have another one here that just says, and we can do an analysis. So they're wondering if we can do an analysis on different fruits and vegetables to tell the best time to pick the fruit, or when it's fully harvested, or when to fully harvest.
Yeah, that's very interesting. Yeah, you definitely could. And there are my lab, not me, but some other folks in my lab did exactly that. And so it's some great papers if you're interested in reading. It's called the Canada's Apple Biodiversity Collection. And it's one of its kind in Canada and Nova Scotia, where we have 1,000 different apple varieties, and there's a lot of studies that are done there.
It's awesome. Ross, I think Ross has a question.
I do. I'm just wondering what kind of commercial applications would your Apple research lead to?
The apples?
Yeah.
Oh, yeah. So that's actually a really interesting question. So the lab that I was working with actually turned into a startup. And so we commercialized, we were, planning to commercialize a lot of the stuff that we developed. So essentially, all it gets to is we find out that these little knobs and that we can change in the genome to improve the food that we see in the table. So like one of the, I don't know if you've heard the stories about purple tomato that's by the Norfolk Ag Sciences in Davison, California. They've essentially what they've done is they've changed some of the things in some of the genes and tomatoes to make it purple. But it's not just to make it look exotic, but it actually has a lot of antioxidants, which is really helpful for the health as well. So this is the type of things that we can do. There's a company down in North Carolina called Pairwise, and they work with a lot of the same stuff. They're looking for all these different targets that you can change in the genome of these fruits. So for example, they work with berries and cherries and things like that. And they're interested in knowing what these knobs are that we can change to improve the fruits. One of the other examples, sorry, I'm really fascinated by this, is
cool.
that same company created a tomato that produces vitamin D. So now you can imagine how useful it is in North America, where we don't get a lot of sunlight. We can eat tomato, which is a staple in every diet, and get vitamin D naturally.
That's awesome.
That's awesome.
Yeah, it's really interesting stuff for sure. And I noticed we have another another question here. I think you really stirred up the chat here. So we have a person who let's see. So somebody's just wondering, sorry, the chat's just flying all over the place on me here. So they're just wondering if this can be forwarded to medical to help pinpoint what people are sensitive to as far as food and scent allergies, or if there's anything there.
Yeah, absolutely. So actually, the supervisor, Sean Miles, whom I work with on apples, actually has this genetic mutation that makes him allergic to apples. So when you bite into there's apple allergy is pretty common. When you bite into an apple, there's like redness and things like that. You can actually do this type of analysis to figure out what is it in that genome that causes that mutation and that phenotype. And so this GWAS is widely used across the domain, right? It's used in healthcare, it's used in livestock, it's used in agriculture. It's a it's a very common and useful tool to see what's happening in the genome. And it's all, and one of the interesting things that I wanted to point out is it's all linear regression. Like it's nothing more than that. It's nothing really complicated. You're essentially taking all these SNPs and you're doing a series of linear regressions, and you're visualizing that. That's all there is to it.
There was another one that was popped my way, and just since you mentioned linear regression, one question that somebody had here is, how has Gen AI changed the work at all? I know it's a hot topic, so I think there's always going to be people wondering those things, and I'm curious too.
Yeah, well, so there was this podcast that I was listening to for a professor who was working with rare diseases, right? So one of the issues with GWAS is that you're able to find things that are really widely out there, right? But those rare variants that are going to be filtered out, you're not going to be able to find with this. And a lot of the, not a lot, rarely there are patients who have very rare diseases, and we need to, get around that and figure out what is exactly is caused by that. And so there's a researcher that, her entire job is to figure out what genetic mutations cause these rare diseases. And so, I was listening to the podcast, and they were talking about the new model that had come out where, ChatGPT's new model that does critical thinking. They've gotten, a little bit more advanced with that, and it actually helps, people think about where to look for, some clues to sort of explore that space, which is really interesting.
Yeah, that's exciting. We've got one more here saying, I'll do two more questions, okay? Then we're gonna wrap it up, okay? So we've got, there's an idea that Sakina suggested, if you can do an analysis on the hybridization to the process of crossbreeding two different plant species or varieties to create a new fruit with combined characteristics from both plant. So I think maybe can you use some of this to predict, what characteristics you might have in the plant?
Oh, yeah, exactly. And so, this type of breeding has traditionally happened, right? Ever since agriculture has modernized, we take two phenotypes we're interested in, two parents, we breed them together, we, get all this progeny, we pick out the ones that are interesting, and you breed them together, and that's how breeding has generally happened. But the challenge in apples is that it takes 7 years for apple to go from seed to fruit. So if you're testing the phenotypes, you have to wait 7 years. That's a long time. That's why tools like GWAS, genome-wide-associated studies, are really important to sort of figure out what the phenotypes are going to be before you get the fruit.
And then there's one last one here. I think this is more a lament of their favorite kind of apple, but Brad asks, what happened to the golden delicious apples? I think Brad can't find them anymore.
I don't know about that. I could swear that I saw Golden Delicious a couple of weeks ago at Walmart, but I don't know.
Hey, good stuff. Good stuff. Well, you know what? I just want to say thank you so much, Tayab. We really appreciate you coming out. This was a super interesting talk. I like how you threaded in the bioinformatics, the data side, as well as tying it back to a real cool product that you guys have put together. So thank you. Before we run, I just want to put one more shout out before we go here. So in addition to all the stuff we're doing, because we have a lot of things on the go, we would appreciate any help you folks might want to give. I know a lot of folks have signed up for projects. We're doing our best to get more projects in the pipeline, but Scott, Ross, myself, and the rest of the leadership team are all constrained by the fact that, we're trying to do a lot of things. And so any support that we might have, anybody who wants to reach out and just jump in to help us with any of the creative or social media support, any of our general event support, or just overall organization and support just for Data for Good in general, just to help us out with various things that need to get done. It would really help out, because then we have a lot more time to go out, canvas for speakers like Tayab, canvas for projects for our members to get involved with. So if you are interested, please feel free to reach out to myself, or Ross, or Scott. I'll throw myself in the chat so that you have it, ryan.tessier@dataforgood.ca, or you can go onto our website, Saskatchewan.dataforgood.ca, and you can kind of sign up on our link. So again, anybody who wants to help out, we really appreciate you getting on board and trying to help us out with finding great speakers like Tayab and exciting new projects. So, but again, just a final thank you to Tayab. We do really appreciate it. So I'll give you a little reaction, a little clap. You know what? Actually, let's all react with tomato just for Tayab. Just a purple tomato emoji down in the chat there. Make sure you guys reach out, and if you guys like to talk, you can. I'm find Tayab on LinkedIn. Adam, bug him. Try to get involved. See how you can learn more about what's going on. Sorry, Tayab, to throw you out there, but I'm sure you guys want to.
No problem.
This is
yeah. This is what I love every day. So yeah, no, happy to chat. And thank you so much, Ryan, and the Data for Good team for organizing this. This is actually really amazing, and I'm really excited to contribute more into Saskatoon. I don't have to drive 3 hours, for the sessions. But no, I'm really excited about the Saskatoon chapter here.
Appreciate that. All right, everybody. Well, have yourselves a great day, and yeah, it's raining over here, so keep dry. See you, everyone.
You guys, bye.